Hello guys, welcome to my channel. Myself Maithili. Today in this video, we are going to see about microbial production of acetic acid. First, what is acetic acid? Acetic acid is a colorless liquid and a weak organic acid. It has sour taste. Acetic acid is also called as ethanolic acid, vinegar, methane carboxylic acid or hydrogen acetate. Vinegar is nothing but 4% of acetic acid. The term vinegar has originated from the French word which means sour wine. Vinegar fermentation involves souring of wine under specific conditions. Production of vinegar is a two-stage process. First, alcohol fermentation. Second, acetic acid fermentation. In alcohol fermentation, yeast enzymes use the sugar to produce alcohol and carbon dioxide. In acetic acid fermentation, gluconobacter and acetobacter converts the ethanol into acetic acid and water. Next, microorganisms involved in acetic acid production. Acetic acid bacteria is the gram-negative bacteria which oxidize sugar or ethanol and produce acetic acid during the fermentation process. Acetic acid bacteria are rod-shaped and obligate aerobes. Microorganisms involved in the acetic acid production are Acetobacter aceti, Gluconobacter oxidans, and Acetomonas species. Two groups of acetic acid bacteria are commonly used for the production of acetic acid. Acetobacter and Gluconobacter are commonly used for the acetic acid fermentation. These organisms oxidize ethanol to acetic acid. Acetic acid production. Commercially, acetic acid is produced by these three methods. First, the Orleans method. Acetic acid bacteria grow slowly as a film over the surface of still liquid. Second, surface fermentation process. Third, submerged fermentation process. Nutrient concentration used in submerged fermentation is generally 5 times greater than surface fermentation process. Orleans method. Old and traditional method, slow but 98% fully continuous process. High-grade vinegar used as a starter culture. Large wooden containers were used in this process. Conversion of acetic acid is done within 5 weeks to 6 weeks. Surface fermentation process. Trickling generator is generally used in this process. The containers were made up of wood. Inner surface lined with wooden shavings. Oxygen is allowed into the container through two routes. Media used for the surface fermentation process are glucose, ammonium phosphate, magnesium sulfate, potassium citrate and potassium chloride and autolyzed yeast. Substrate used are ethanol, wine, whey, molasses, fermented grains, malt and rice. Submerged fermentation process. Production rate is 10 times higher per cubic meter than the surface fermentation process. The starting material 100 ml of 5% ethanol is used in the process to get 7 to 10 gram of acetic acid per 100 ml. Fermentation is done in stainless steel and they are stirred from the bottom. Fermentation up to 35 hours at 40 degrees Celsius temperature. Heat exchanger is provided to maintain the temperature. Yield. The yield of acetic acid is about 98% in fully continuous process. Recovery. The vinegar produced by submerged fermentation is turbid due to the presence of bacteria. This turbid solution is clarified by filtration. The plate filters and filter aids are used. Decolorized if required. Application of acetic acid. Acetic acid are used for the preparation of perfumes and used to make dyes like indigo, used in production of rubber, synthetic fibers and plastics, used in vinegar production, used for pickling of raw vegetables. Thanks for watching this video.